So then our last biological molecule are the proteins. So now the proteins have the building blocks called amino acids. So there are 20 amino acids, but they all have the same base structure, and then only the side chain differs. So it starts with a carbon in the middle, and there's a hydrogen on it. Then there is an amine group. So an N with two H's on that side is what we call an amine group. Then on the other side, there is a C, double bonded O, OH, and that is a carboxylic acid group. And that's how it gets its name, because it's an amino acid, because it has the amine group and the carboxylic acid. And then the R is our side chain, and the side chain differs for each amino acid. So that's why we represent it with an R, because it's different for all 20 amino acids and it changes. But you don't have to know all 20. You just have to know the basic structure, and if they want a specific amino acid, they will tell you what the side chain is. So this is always going to be what all the amino acids are made of. And now, if those amino acids need to be joined together, they're going to line up. So we will have one amino acid next to another one, with the carboxylic acid group lining up with the amine group. So now, just like before, with all the other molecules, two H's and an O can come off as water, because when we join things together, it's a condensation reaction because we make water. And now the amino acids are joined together. So we have our carbon in the middle, the carboxylic acid side, but now that carbon gets joined to the nitrogen on the other molecule, which has its one hydrogen left. And then we can fill in the rest of the molecule on that side and on that side. So this bond there that gets made between the C with the double bonded O and the N with the H, that bond we call a peptide bond. So between amino acids, you make a peptide bond between the carbon and the nitrogen. So the carbohydrates and the fats had the oxygen in the bonds to make the glycosidic and the ester bonds. But now for a protein, there's no oxygen in the bond, and we make a peptide bond. So two amino acids together we call a dipeptide. Many together make a polypeptide. So this is just two, but then we can join another one to that side, remove the OH and the H, coming off as water, to the next one, to another one. Okay, and because we are making them and water is coming off, going that way, it's condensation. And if I wanted to break them by adding water back, it would be hydrolysis. So now, to make a protein what it is, we can't just only get a single strand of amino acids. What we have are called levels of structure to a protein. And the protein levels of structure are as follows. We start off with a primary structure. So a primary structure is the sequence of amino acids. So that gets decided by the DNA code, and it's just what amino acids there are all joined together to make up my long polypeptide. So each one of those is all different, And that's the sequence of amino acids, all in a straight line, all with peptide bonds. Once we have the primary structure with the sequence, we then get a secondary structure. So in a secondary structure, we create hydrogen bonds. So a hydrogen bond forms between the amine groups and the carboxylic acid groups on all these amino acids, and it starts to arrange itself in a helical shape. So it starts to form a helix with hydrogen bonds forming between our chains. And we call that an alpha helix, or it could form a beta pleated sheet in which they form little pleats around each other. So that's a secondary structure with hydrogen bonds in an alpha helix or a beta pleated sheet. The tertiary structure, which comes next, is where this alpha helix and beta pleated sheets and that folding starts to create a 3D arrangement. So they start to form even more bonds with each other. And that's how we get this giant 3D structure that has a specific shape based on the sequence of amino acids. So now the 3D structure in the tertiary bonding consists of hydrogen bonds, ionic bonds, disulfide bonds, and hydrophobic interactions. So those are the 4 different kinds of bonding that we can find in this 3D structure. So our hydrogen bonds, same as before, with things with oxygen and hydrogen, ionic as well as nitrogen. Ionic bonds with charges. Disulfide bonds contain sulfur, so that's usually only cysteine. And then hydrophobic interactions are all the side chains that hate water. So all of these bonds all occur between the R groups of each of those amino acids, and that creates this big 3D structure. And then a quaternary structure is if I take more than one of these polypeptides and I join them together. So I can have my one polypeptide and it gets held in place with another one. So more than one together make a quaternary structure. So not every protein has a quaternary structure, but if they do, it means there's multiple polypeptides all put together.